Wagwan, my GCSE science students, welcome back to another video. In today's video, we're going to be looking at chapter 5, communicable diseases, okay? And in this chapter, you need to know about health and disease, pathogen and disease, preventing infection, so looking at the work of Ignaz Semmelweis and washing hands, all of that, viral and bacterial disease, fungi and protists, the human defense responses. And then for the triple scientists, we're going to be looking at growing bacteria in the lab, preventing bacterial growth, and plant diseases and defenses for triple science. Okay, so here goes. First thing that we need to know for this chapter is what exactly is health. Now, this is a definition that you should be writing on your exam. So the health is a physical and mental well-being. Is a state of physical and mental well-being. Okay, so it's not only just an absence of disease, and it's not only your physical. You need to be talking about your mental well-being. Too easy. All right, now there are two different types of diseases that you need to know about. For this chapter, you have your communicable disease, which are the infectious ones. All right, and then you have your non-communicable diseases, which are the ones which are not infectious. So their communicable diseases are caused by pathogens. All right, and when we talk about pathogens, we are talking about things like bacteria, viruses, protists, and fungi. Okay, good. And also some examples of that would be like tuberculosis and flu. But when we're talking about non-communicable diseases, these are diseases which cannot be transmitted from one person to another. Okay, so we're talking about diseases such as like heart disease, cancer, arthritis, diabetes, diseases like those. They're not infectious. They're not spread by a pathogen. Okay, good. So communicable diseases are the ones that can be caught. Okay, so they are spread or they are transmitted by microorganisms, and they can pass from one person to the next person. Okay, and as we mentioned before, these are your fungi, your protists, your bacteria, your viruses. Okay, and here's some pictures just to help you to see what they look like under the microscope. Too easy. All right, now, do you think anything else affects your health? Okay, and your answer to that should be an obvious yes. All right, so there are other factors which affects your health. Your diet can affect your health. How stressed you are can affect your health. Life situations can affect your health, believe it or not. All right, and now we're going to look at how exactly these factors affect your health. Okay, and what diseases can be caused from this. So when you think about diet, I mean, this is so important to your health. All right, not eating enough, all right, or not eating the right set of nutrients can lead to certain deficiency diseases, certain starvation diseases. Okay, eating too much of the wrong food especially can lead to certain diseases such as type 2 diabetes, some cancers, and obesity. Okay, and you have some pictures here. All right, so you can see obesity, you can also see rickets, someone who is anorexic, not eating enough. So diet is absolutely important, which is why the government is doing so much work with the food that we eat. Okay, all right, good. Another thing that can cause ill health or diseases is stress. Okay, so I mean, we are going to be faced with stress, so certain amount of stress we are going to face that. However, too much stress can increase your risk of so many diseases, heart diseases, cancer, mental health problems. As a matter of fact, stress releases a hormone which is called cortisol, which can have severe impact on your body. Okay, good. Too easy. Next up, your life situation. All right, this has been going on since the beginning of time. Wherever you live, your gender, believe it or not. So for example, black men are more predisposed to having prostate cancer. Okay, so your financial status again can affect whether you have certain diseases, believe it or not, or if you get sick, how do you get treatment? All right, all of that can affect you as well. Okay, your ethnic group, as I just mentioned before, black men are more predisposed for having prostate cancer when compared to our white counterparts. Okay, and there are lots of diseases out there which are more prominent in different ethnic groups. Okay, healthcare provided. all right, that can affect us, how many children you have, your local sewage and your rubbish disposal, air pollution, all of that can affect whether you have diseases or not. Make sure if you're asked, you're able to just explain each one, okay? So communicable disease on the other end is caused by pathogens, okay? And the definition for that word, pathogens, is any microorganism which can cause an infectious disease. That's the definition for pathogen. Write that down on your flashcard straight away. Too easy, okay? Now, how are these pathogens spread? Now, you have 3 pictures there to help you. So it can pass through the air. We've lived through one, which is called COVID, direct contact, or through water. Pathogens can be spread in any one of these 3 ways. Too easy, okay? So how exactly do pathogens cause disease? So when you think about something like bacteria, that causes disease by the fact that they divide quite quickly through binary fission, and then they produce toxins which are poisonous, okay? And these toxins can then directly damage your cells. Then you also have viruses, okay? So what the viruses do, they live and they reproduce inside the cells, and then they damage the cells. And then because they reproduce, you're going to have several other viruses which then attack other cells, okay? So you can see a picture of the virus there. All right, that's what it would look like, okay? And it goes inside your cell, it divides and then spreads to other cells. Now, types of pathogens, there are 4 that you need to remember. I've been saying it since the start of the video: bacteria, viruses, protists, and fungi. Make sure that you know the 4 of those, okay? And you also now need to know each or name type of disease. This is coming straight from the spec. So you need to know that for virus, there are 4 that you need. There are 3 named viruses that the specification wants you to know. Okay, measles, HIV, and the tobacco mosaic virus. Okay, this is for the AQA board. All right. So in terms of symptoms, you can see that there. All right. You've seen people with measles before, I assume. All right, so it's that fever, that red rash, and it can lead to actually damaging your brain, and sometimes can be fatal as well. It can cause death, okay? Now, it is spread through droplets, all right, from coughs and sneezes, and it can be treated by, you can be vaccinated against it. So there's vaccines out there, which is called MMR. So there's an MMR, measles, mumps, and rubella vaccine out there, all right? Or the person who has the disease can also be isolated. HIV or AIDS, all right, symptoms will vary, all right, but it tends to be like flu-like symptoms, but your body can't fight that off and can't deal with those types of simple infection, so it's reoccurring. It is spread through direct contact, sexual contact, or exchange of bodily fluids, okay? And how is it controlled? It is controlled through the use of antiretroviral drugs or minimizing sexual contact, okay? And then you have your tobacco mosaic virus, all right? So there's a mosaic pattern, okay, of discoloration on the leaf of the plant. All right, so they're not able to photosynthesize. They cannot carry out photosynthesis because the leaf is now discolored, normally yellow, okay? And how is it spread? Direct contact. Well, contact between the diseased plant material and healthy plant material, or insects can act as vectors by taking the virus from one plant to another. Or if a farmer uses tools on a plant which contains a tobacco mosaic virus and then uses it on another plant, that's how it's then transferred, okay? Now, how is it treated? All right, the only way of treating is to basically isolate the plant, the infected plant, all right? Or you could then grow tobacco mosaic virus resistant plants, all right? You can also practice good hygiene and pest control. So you could get, make sure that your tools are not being cross-contaminated with plants which have tobacco mosaic virus, okay? You can use this to fill in your flashcard. Too easy. Then, let's look at the bacteria. So for bacteria, the exam board wants you to know about salmonella and gonorrhea, okay? So for salmonella, symptoms include abdominal cramps, vomiting. It's basically fever, diarrhea. How is it spread? Bacteria found in raw meat, poultry, and eggs. So that's why these things need to be cooked properly. How is it treated? Well, in the UK, they currently vaccinate to reduce the spread, but there is no real treatment. It's just naturally there. And then gonorrhea, in terms of symptoms, there's a thick yellow or green discharge rather from the reproductive organ, the vagina and the penis, and it could be pain when urinating. All right, direct sex contact, and it is treated using antibiotic because it's a bacteria, or you're wearing condoms during sexual contact to reduce the spread. All right, now as it relates to the fungi and the protists, you need to know about the the rose black spot. Come on, GCSE science was. So symptoms include the purple or black spots which develop on leaves, all right, which can then turn yellow and then they fall off, and then it is spread through wind and water, and it is treated using fungicides. Okay, as it relates to malaria, symptoms is a reoccurring episodes of fever, and this can be deadly. Okay, it is spread through mosquitoes, all right, which is a vector for the protists, good, which causes malaria, and then this is treated using anti-malarial drugs or preventing the vectors from breeding. So that would include areas where it has a lot of water, collecting like pots and pans, make sure that that area is kept free from water so that the mosquitoes don't have a breeding ground. So no mosquitoes, no vector, therefore reduction in malaria. Okay, good. Semmelweis, he was very important to the work that goes into washing our hands, okay? And not only washing our hands, but washing your hands in a hospital setting. So what he found out, he saw that a doctor who was working in the morgue dealing with a corpse, that doctor died from similar symptoms to the corpse that he was working on after cutting himself, okay? As I mentioned before, he insisted that hands needed to be washed before delivering babies. And after he insisted on that, he realized that fewer mothers were dying. So, during the mid-1850s, so long time ago, right? Many women in the hospitals, they were dying from childbed fever few days after giving birth. Now, this was very puzzling to Ignaz Semmelweis, right? So no one was knowing what was causing it at the time. Okay. However, Ignaz Semmelweis, he realized that the women who were seen by doctor or a midwife, there were discrepancies, there were differences in the death rate. And we're going to look at a graph with that. So the doctors, they were doing work inside the morgue where they kept the dead bodies. Okay. So they were working on corpses. Okay, and the midwives, they did not work with corpses. So that's the first thing that you need to realize. And what he found out is that the women that were seen by the doctor, they were more likely to die of childbed fever. So he's thinking that something is coming from those corpses. Okay, so that could be some sort of bacteria taken from the corpses and then taken to the women who were giving birth. Okay, so look at the data here. So this graph in the pink here, it shows the mortality rate, the death rate of those who were working in the morgue. So these are by the doctors, and this is by the midwives. So you can see the difference in the number of deaths. Okay, so based on that, Ignaz Semmelweis realized that the doctors had something on their hand. He didn't know what it was at the time, all right, but they were carrying around bacteria. But I'm going to put that in quotation because they didn't know what that was at the time. So he asked for them to wash their hands when leaving the corpses to deliver baby. However, this did not go down well, believe it or not. Washing hands or asking doctors to wash their hands after touching dead bodies did not go down well. And why did it not go down well? It didn't go down well because, one, you're making a suggestion that the doctors are the ones that's causing the death of these mothers. And that's ego when you think about it. Doctors, I'm not going to wash my hand. Who are you to tell me to wash my hand? Okay, good. Also, he did not have enough evidence to back up what he's saying. Science is about evidence. They could not see the bacteria on their hand. Okay, so he's asking them to wash something that they could not see. Remember, this is in the 1850s. All right, and sadly, because of this, Semmelweis went crazy, and in 1865 he was actually admitted into an asylum where he died at age 47 from a blood infection. However, because of the work that Ignaz Semmelweis found out about washing hands, this is now everyday practice. Okay, this is now everyday practice. So make sure you have an idea about the work of Ignaz Semmelweis. So this allowed for the work of hygiene, hand washing, disinfecting kitchen surfaces and toilets, etc. Keeping away meat away from food, raw meat from food. And when you cough or sneeze, make sure that you're using a handkerchief or tissue and wash your hands. Okay, he pioneered the work for this. All right. Also, if someone is sick, you can isolate that individual. If someone has something like cholera or Ebola, or during even COVID, we were asked to isolate. Okay, and this is only a couple of years ago. All right. This method is also used for plants as well that can be removed and destroyed easily. You ask those plants to, well, you don't ask them, but you remove them. Okay, good. All right. Also, in terms of preventing diseases, you can destroy or control the vectors. So, for example, we spoke about earlier the mosquito and malaria. In order to reduce that transfer, you can destroy the vectors or control the vectors. Rats, for example, aphids that we're going to look at later. All right, those can transfer certain or carry certain pathogens. All right, you can destroy the vectors and therefore prevent the spread of these diseases. Now, there are certain ethical arguments behind this. For example, killing the rats or killing flies that can affect the food chain, the food web, and therefore have several implications down later. But there's no right, there's no wrong. In the exam, if asked, make sure that you have arguments for and against the destroying or controlling vectors. Okay. Good. Vaccination also is a way of preventing diseases. Okay, so what happens here? You place a small amount of the harmless pathogen inside your body that will then trigger some sort of response, and your body now has certain memory cells. We will look at this later in the next chapter. All right, good. And then you cannot protect plants against diseases because they have no immune system. So we can't vaccinate plants. Okay. Now, next up, we're going to be looking at the human defense system. All right. So any opening or break in the skin, those are entry points for diseases. Okay. Which is why cuts have to be treated so carefully. All right. So more pathogens in your body, the more chance that you will get some sort of infectious disease. Now, in order to prevent this, the body has several defenses in place. Now, the first defense is the skin defense. All right. So the skin just acts as a barrier, preventing microorganisms from going in. The skin also produces antimicrobial secretions, which is going to kill certain microbes. And also, your skin is also covered with microorganisms that will keep it healthy. All right. Another way that the body protects itself is through the respiratory system. Okay. So your nose has lots of hairs, which will and it also produces mucus, which will trap bacteria. Okay. Good. So this will trap any bacteria, any pathogen. Your trachea also secretes mucus. So this here is your trachea. All right, mucus is secreted there, which will trap again any bacteria. There's also cilia, which is there, which then moves any bacteria which is there so if that's your cilia and that's your mucus with bacteria, okay, this will move left to right and then shift the bacteria out of the system, okay? Your digestive system is also another way that the body protects itself, believe it or not. So the stomach produces an acid which will kill any pathogen, all right? And then vomiting or diarrhea will get rid of any pathogen, which is why if you eat certain type of food and it has certain bacteria on it, you tend to bring that up, or you have diarrhea to remove it so that it doesn't stay in your body and multiply. Too easy, okay? You also have the immune system, okay? Now, this immune system, if the first line of defense is breached, which is your skin, the immune system then takes over, all right? So your white blood cell come into play in order to defend the body. Now, the very first way in which the white blood cell defends the body is through eating them and destroying them in a process called phagocytosis. So what you can see here, the white blood cell is engulfing the bacteria. It will digest it, eat it up, and get rid of it. Phagocytosis, okay? So these types of white blood cells, they are called phagocytes. Okay, too easy. All right, then you have another type of white blood cells. They don't carry out phagocytosis. What they do, they produce a unique type of antibody, okay? So here's your white blood cell, all right? Here's the antibody, which is complementary to the antigen on the pathogen, okay? So it produces the antibody, which attaches itself to it. And once the antibody is there, made quickly, right? It then attaches it. There's a marker there. Another white blood cell comes in and destroys it, okay? Which is why if you get that bacteria again in your system, the antibody is already there and in larger amounts to defend your body, okay? Finally, there's some white blood cells which produces antitoxins, okay? And these counteract the toxins which are produced by pathogen. So your pathogen will release toxin. Antitoxins are then produced, which will counteract it and prevent you from getting sick. Okay, so those are the three ways in which white blood cell can defend your body. Okay, now if you are combined science, you can end the video here. I'm going to continue for my triple scientist. Okay. Good. So for triple science, you need to know how to culture microorganisms. This is one of your core practical. Now, why is it that it is useful? Is because scientists need to know how to culture bacteria, because we can then study the bacteria inside the lab. So the more we can culture them or produce them, the more that we can be able to study them. We need to grow them in large numbers so that we can study them, and we call this a colony of bacteria. Okay, we can also use it to investigate which disinfectant or which antibiotic is good at destroying these diseases. Okay, now you need to know the steps as to how to culture the bacteria. Now, in the first step, you disinfect your area. Before you begin, disinfect and clean the area, okay? And the reason for this is so that you reduce the risk of contamination, all right? Your next up, you're gonna have a loop, which that you make sure that that loop is sterilized over the Bunsen burner. This is to kill any bacteria that may be on the loop already, okay? You're doing all of these as aseptic techniques, okay? So you need to know the aseptic technique and why you're doing, okay? Then you're going to collect your bacteria, okay? So your bacteria is in some sort of conical flask. That loop that you had, you're going to dip that into the bacteria here, and that process is called inoculating the loop, okay? You then would have some sort of agar gel, okay, prepared in, right? So you have agar gel here. You're going to inoculate the agar gel by making a Z shape. Good. You then need to cover or seal that agar plate. All right. Good. But do not seal all the way around, because the bacteria, some of them will need some oxygen to grow. Okay. Unless you have some anaerobic bacteria. Okay. Good. Now, after you have that petri dish with the agar and the bacteria which has been inoculated, you need to store it upside down. Very important. And the reason for that is to prevent condensation from forming on the agar surface. You leave it for some time, all right? And sometimes you actually place it in a warm oven at the right temperature, all right? 25 degrees for 48 hours. And the reason for doing this, we don't want to put it past 25 degrees because you don't want to kill them, okay? And to reduce the risk of growing pathogens that might be harmful as well, because pathogens that can live above certain temperature, they're quite harmful. And then you disinfect the area again, all right, to kill any bacteria that may be spilled on your desk after you've taken out and observed it, okay? Now, after you've prepared your bacteria, you can then look at the next part of the practical in which you're going to be investigating the disinfectants on those microbes, okay? So you're preparing your agar, your bottle, so you flame the area, all right, aseptic technique. Good. Then you're going to pour your agar in a sterile plate, all right? So now, when the agar gel is ready, we now add our bacteria in with that Z shape that we discussed before, okay? Now you're gonna have your forceps to collect your paper disc, which has been soaked in some sort of disinfectant or antibiotic, okay? So now you have your different disinfectants here. One of them is going to be a controlled, okay? Think about why would you need some sort of controlled. All right, so you have a disinfectant 1, 2, 3, 4. All right, good. And then if you notice, there's some bits or some area around where no bacteria is growing, okay? It's called the zone of exclusion. All right, good. You're going to measure the diameter of each disc where you have no bacterial growth. So you have your zone of exclusion. The zone of exclusion. All right, good. And then you can also work out the area where there's no bacterial growth. So you can use pi r squared, and then you draw up your table and then you fill in your necessary information. So here's an idea as to what the table would look like. All right, so you have your clear zone, all right, your radius, and then you have your area, you have your different antiseptic use here. So in this practical, you need to know the independent, the dependent, and the controlled. So your independent variable is that you're using different type of antibiotic or antiseptic. Your dependent variable is your zone of inhibition, okay? So you're gonna have to your zone of inhibition or your zone of exclusion, and also your control would be the size of the disc. So you need to ensure that you're using the same size disc and the time that you soak the antibiotic in for. All right, now why I mentioned why we use water, right, is used as a control so that you can have some sort of comparison or confirm that there's no bacterial growth, or the no bacterial growth is due to the effect of the disinfectant that you have there, okay? Now, also for triple science, you need to look at the plant diseases, okay? So, as it relates to plant diseases, there are 4 plant diseases that you need to know. So, you need to know about the tobacco mosaic virus, the black spot virus, aphids, which are acting as a pest, and you need to know about the mineral deficiency diseases, okay? Good. So TMV is caused by a virus. The black spot is caused from a fungus. All right. So let's have a look at that. So aphid. Here's a picture of the aphid, so you can have a look at what is happening here. Now, aphid, we need to know what problems can be caused and how exactly we destroy them. So for the aphid, what they do is that they penetrate into the phloem vessel. And remember that the phloem is carrying around the glucose, which is made from photosynthesis, okay? And then they penetrate that and feed on it, okay? Now remember, if you're taking away the sugar, which is required for respiration, made from photosynthesis, this is going to damage and weaken the plant, okay? They can also act as vectors, so they can transfer viruses from one point on a plant to another plant itself, okay? Now, how do we destroy aphid? You can use pesticide, You can also have them in an enclosed space with some sort of biological pest. All right, so you can get something which will eat the aphids, such as the ladybirds. Okay, so they can eat the aphids. You can see what's happening there. Good. Next up, you need to think about the deficiencies that plants might have. So plants need mineral ions and water to grow. Now, if they don't have enough of those mineral ions, that can stunt their growth. They can have different appearances. They will not function optimally. Okay, so let's look at the first deficiency. So nitrate, very, very, very important for protein synthesis. It's used to make protein, and therefore, if you don't have enough, the plants can't grow. Okay, which therefore means that the plants can't carry out photosynthesis. Protein synthesis rather, is going to stunt their growth, and it's therefore going to reduce the whatever yield that they're making. So if they're producing fruit, you're going to have less of those fruit. All right, so for magnesium deficiency, have a look at the leaves there. Okay, so you can see the one on the right here that has some sort of magnesium deficiency. Okay, because of the yellow in there, because magnesium is an integral part for making chlorophyll. Okay, so no magnesium, no chlorophyll, therefore it will show up as being yellow. All right, so not enough chlorophyll made, leaves become yellow, growth becomes stunted, and therefore you're going to make less glucose. All right, you also need to know that the yellowing of leaf is referred to as chlorosis. Okay, good. Now, if you have a garden, how would you go about detecting if something is wrong in that garden or any plant diseases? Okay, look for stunted growth. You're going to look for spots on the leaves. Okay, so you might have some sort of black spot fungus, areas of decay or rot. All right, how is it growing, or growths rather? If you have any stems or leaves which is not formed the way they should. Discoloration, so do you have any sort of yellowing? It's possibly chlorosis or TMV. All right, and if you have the presence of pests. Okay, and now, so depending on what you are observing, these are the different possibilities of diseases which are happening to the plants. You need to know that, triple scientists. Okay, so here are some. Have a look and see if you can identify what these diseases are. All right, good. Now, how do you identify what the cause is as a farmer? You can refer to a garden manual or website. All right, you can ask for an expert to come in, visit, and take DNA analysis of the pathogen found. All right, you can also have testing kits with certain monoclonal antibodies which will identify certain pathogens. All right, and we're going to look at monoclonal antibodies later on. Now, plants also have their barrier system in place. Plants have their physical barriers, they have their chemical barriers, and they also have their mechanical barriers. And we're going to look now at each of these, all right, and some examples. So for the physical barriers, plants have their cellulose walls in place, first and foremost, which gives them strength and helps to prevent invasion from certain pathogens. Okay, good. They also have a tough waxy cuticle which prevents pathogens from coming in. They also have barks on trees, which is that layer of dead cell, and it acts as protection. And also leaves, they can fall off during if it becomes ill or infected, they will fall off just like in winter. All right, good. Plants also have their chemical barriers. So believe it or not, plants also produce certain antibacterial chemicals. And these chemicals, actually, we've been using them for certain treatment of diseases or even an alternative to antibiotics. That's something that we're looking at. So foxgloves, for example, is used in the treatment of certain heart issues. Okay. But foxgloves, they produce poisons to prevent animals from eating them. Okay, you also have your mechanical barriers. So thorns, which are unpleasant to eat for if animals come to eat them. You also have hairy stems, which is stopping larger animals from coming to try and eat them. All right, you can also have it droops up or it curls up, which then when you touch it, so animals will become frightened when they get near to it. Or some of them also do a mimicry, okay? So they're able to they're able to droop, my bad, to mimic unhealthy plants to stop animals eating them, all right? Now, that comes to the end of chapter 5. All right, please remember to hit like. Please remember to share my content with your friends. And remember, too easy. I'm out.